Exercise 10 is a series of experiments to help you identify a bacteria. Each bacteria has its own set of characteristics, what it will digest, how it reacts when you expose it to certain things, whether it is gram-positive or gram-negative. Push comes to shove, these tests are to help you narrow the field of which bacteria you could possibly have. So the purpose of this series of experiments. Biochemical characteristics are useful in differentiating between various strains of bacteria and allow scientists to detect many unique differences between the bacteria and aid in their identification. One of the considerations that you have to have is your cultures have to be pure. Last week we did pure culture where we had a mixture of bacteria and we streaked it on a plate to try and get individual colonies. In this case, if you were in the lab, I would give you pure bacteria. It wouldn't be a mixture anymore. So what you are using is pure culture. The first series of tests we are going to do is for acid formation to prove that my bacteria are eating the sugar inside of a tube. And then gas evaluation. When they're doing this, do they produce gas? Fermentation tubes. So in a fermentation tubes, you have a test tube that has a screw cap, and you've got this itty bitty little tube that's called a Durham tube. And it is a test tube, but it's very tiny. You put this in upside down. So the mouth of the tube is facing down toward the bottom. You put this in, you put your solution in here, and you cap it closed. One of the things that you will do is you will make sure that there is no air inside of that tube, inside of the Durham tube, the smaller tube. When you're done, it looks like this. You can hear the tube that's still in there. There's no gas in it right now. So why do we go through that trouble? Well, when we have sugars in these tubes, what we're looking to see is what specific sugar do these bacteria eat. Remember that fermentation is one of the ways our bacteria can use sugar to get themselves energy. We have 4 different types of tubes like this. We have lactose, sucrose, maltose, and dextrose. All 4 of these tubes have a completely different sugar. If the bacteria doesn't have the specific enzymes it needs for that specific sugar, then nothing should happen in the tube. It should stay exactly like this bacteria-less tube. But if it does start to ferment the bacteria, one of the things that's going to happen is it's going to form acid. As it forms acid, the red pH indicator phenol that's in this tube is going to start to lighten. As it becomes acidic, it goes yellow. So you start with a tube that looks something like this. If the bacteria is really, really good at using the sugar that's in the tube, it'll end up a lighter color, meaning that I ate the sugar, I made acid. Now, the gas part of this, that's what the Durham tube is for. Since the Durham tube is upside down and your bacteria are most likely at the bottom of this test tube down here, as they produce gas and that Durham tube is upside down, the bubbles of the gas get caught in the Durham tube. It's a way to prove gas was made. Why? Because when you started, you made sure there was no air inside of that tube. You have this Durham tube in this example that has an air bubble. That means not only did I use the sugar in here, but I also formed gas when I was doing it. So whatever bacteria A is specifically, it actually did both. It used the sugar, let's say it was sucrose, so it used the sucrose, And while doing it, it produced a gas. Not all bacteria can do this to the same extent. So if you look, there is a range of colors here, going from nothing in the tube to bright yellow. When you have this middle range here, you would say that some acid was produced. Some acid was produced here. Some acid was definitely produced here because it's not the same color necessarily as the original. When you're noting this in your lab manual, you would kind of grade how much acid was produced. Is it a little bit of acid, middle ground acid, or a lot of acid? With just a teeny tiny bit, you would put a lowercase a. With orange, I would say probably a lowercase a with a plus. And then with bright yellow would be a capital A. In the past, I've had this phenol red, that's our pH indicator, turn green. I asked the company what was happening, and they couldn't really give me an answer, but it does happen, so that's kind of interesting. As far as the Durham tube goes, you can kind of also grade that. Either you have a little teeny tiny bubble up here, which would be a little bit of gas. You could have this much gas, which is, you know, a good amount. Or sometimes this whole entire thing is almost full, in which case you have massive gas production. The second test we are going to do is to determine if your bacteria can use starch. For the most part, for the last, I don't know how many weeks, the only plate I ever showed you was a nutrient auger plate, which you can see it says Na. However, we do have other plates. This one is a starch plate. And you know that because it says STAR, it says star, meaning starch. Again, certain bacteria have the ability to use starch as an energy source or as a fuel source to make ATP, but some don't. Starch itself, when you combine it with iodine, turns jet black. If you've got bacteria on a plate and, excuse me, you put iodine on it, the plate, if there is no starch digestion, turns black. However, what color is iodine? It's kind of an amber brown, right? If there is starch digestion next to where your sample is, it'll turn a brown color. Now, I want you to note something. In your lab manual, there is a typo. It says that it turns black when it has starch digestion and turns black when it doesn't. That is not right. Starch digestion happens, it's brown. No starch digestion, black. Literally, you can get starch and cornstarch and put iodine on it and it's black. Looking at this example, I've got one type of bacteria, clearly it's there, that when I add iodine to the plate, it's black. This means that E. coli can't do starch digestion. Since there was starch right next to these colonies, you're done. However, if you look at this example over here, you'll notice that the area around it is actually amber, the color of the iodine. So this particular bacteria, whatever it may be, is actually able to digest starch. Again, why are we doing this? We get more information about the bacteria and the characteristics it has. It makes it easier to identify. It makes it easier to narrow the field like a detective of here's a clue, here's a clue, here's a clue.
Little thing that I forgot to mention. With the starch digestion, remember that your bacteria are sitting on top of your auger. and they can only digest so much starch along that surface. If the agar is very thick in that plate, once that iodine settles and starts to sink into the agar past where your bacteria have actually been able to digest the starch, that agar is going to turn black. Now, what does that mean? It means that once you put the iodine on, I'd say within 2 to 3 minutes, you need to look and see what your results are. Most of the time myself, when I'm doing this experiment, I will put the iodine and give it maybe 10-15 seconds and automatically look to see what my results are. The time frame is not that long to prove what you've got, starch digestion or not. But as I said, if you let that iodine sit and actually soak into the plate, you're going to end up with a negative result that actually isn't negative. In other words, don't let it sit for 10 minutes and then go look.
The next test we're going to do is a catalase test. In Chapter 7, we discussed the danger of oxygen to any type of bacteria. If they didn't have specific enzymes, superoxide dismutase and catalase, then if they got exposed to oxygen, it would most likely kill them because they had no enzymes to detoxify this toxic oxygen. With the catalase test, we're testing the bacteria for catalase specifically. Now, what did catalase do? Catalase took hydrogen peroxide and it broke it down into oxygen, which is a gas, and water. If you put hydrogen peroxide on your bacteria and you see bubbles, that's automatic positive for having catalase as one of the enzymes inside of that bacteria. It's breaking that hydrogen peroxide you just put on it and making oxygen and water, so you see bubbles. So for this specific test, it's either positive for catalase or it's negative. If you don't see any bubbles at all, then you just kill the bacteria, but it's negative. The last biochemical test you are going to do is the motility, iron sulfide, and indole determination. You are going to use another tube, except in this case, instead of having liquid, this is a solid, just like what we have in plates, except it's a different recipe. So inside of here, we have SIM media, S-I-M, SIM. SIM media is specifically used to look for iron sulfide, indole production, and motility of your bacteria. This is also the one time during lab you will use an inoculating needle instead of the loop that we're all familiar with. So you've got this straight needle and you're going to get bacteria off a plate and literally stab this into this agar. You're not moving your needle around. You want one good straight stab and you pull it out of the media or the SIM agar. Why? The reason that you want just one stab is for the motility test. In the motility test, when you stab into the agar, if the only bacteria that is there is in that stab because it can't move anywhere, you'll just see the stab line. You will not see anything else. However, if you have motility from that stab, you will see cloudiness in the agar. So this is a non-motile bacteria. You can see that the SIM has no cloudiness around that stab. That means the bacteria you put in this stab is still in the stab in the agar. However, if you look at this one, that's a straight stab. This was a straight stab, but see all of this cloudiness around it? You can still see where you stabbed into the agar, but all of this cloudiness means that the bacteria actually moved away from where that stab was into the space where the agar is. Something that's interesting, this is a very easy way to prove motility in your bacteria. Hanging drop is another way that we can prove this, but it's actually more of a challenge to do it that way because you have to have new bacteria, you have to make shadow. There's a lot more involved with that compared to this. Within the recipe of the agar, we have chemicals, when exposed to iron sulfide, turn black. So if you stab this, leave it for 48 hours in the incubator, and you come back and you see black inside of the tube, it's positive for iron sulfide production. If it isn't, no iron sulfide was produced. Finally, the last test we're going to do is to determine if the bacteria make indole. Indole is pretty simple as far as tests go. There's a chemical that we have called Kovacs reagent. It's very reactive. You always wear gloves. You are going to use a dropper, just a regular dropper. You're going to collect some of the Kovacs and you're going to put 10 drops on top of your sample. If you see no change, if it still stays kind of clear yellow, then no indole was produced. However, if you see a cherry red color on top of your sim media, that means that the bacteria that you used to stab this sim tube actually makes indole. These tests are, again, a way to narrow down which bacteria you have, because every bacteria is different as far as what it can digest, what it can't digest, what it can react to, like the hydrogen peroxide, what it doesn't. Is it motile? Is it not motile? Is it able to produce indole? Is it not? All of these clues will help you when it comes to your unknown bacteria at the end of the semester or toward the end of the semester.